All right, now we're going to look at the pancreas. Now, the pancreas is interesting because it has exocrine and endocrine functions. So it has both exocrine and endocrine cells. It's a triangular gland. It's located kind of posterior, partially behind the stomach. The exocrine cells are called acinar cells or acinous cells. The exocrine cells produce enzyme-rich pancreatic juice, and that is going to be used for digestion. So we'll get to the digestive system. We'll talk more about this exocrine function for the pancreas, but let's turn our attention to the endocrine cells. The endocrine cells are known as pancreatic islets or islets of Langerhans. They do contain alpha and beta cells. Now, alpha cells produce glucagon, while beta cells produce insulin. Now, insulin decreases the amount of glucose in the blood, so it's called the hypoglycemic hormone, whereas glucagon increases the amount of glucose in the blood, so it's called the hyperglycemic hormone. Also, you do need to know which cells produce which hormone. So I always just remember that alpha cells produce glucagon because there's an A in it, and then it has to be beta cells for the other one, even though it'd be great if there was a B in insulin. Now let's look at figure 15.16. This is a photomicrograph of pancreatic tissue. We're seeing both the pancreatic acinar cells, so the exocrine cells, and the endocrine cells. So here we have the endocrine cells, so alpha and beta. Don't worry, you don't have to know the difference necessarily between the two in a histomicrograph. But again, remember, alpha cells produce glucagon, beta cells produce insulin. We also want to know the function of glucagon and insulin. So these are the hormones produced by the pancreas, and glucagon is extremely potent as a hyperglycemic agent. So hyperglycemic, that means increases in glucose or sugar. So glucagon definitely increases blood glucose levels. And we're going to see the production of glucagon from the pancreas triggered by a decrease in blood glucose levels, which makes sense. If that's our stimulus, the result is glucagon secretion, which will increase or reverse that initial trigger. So rising amino acid levels or sympathetic nervous system stimulation can also cause the release of glucagon from the pancreas. We're going to see again that glucagon raises blood glucose levels. So I'll say that multiple times just to make sure it's very clear. But let's talk about how we're going to raise blood glucose levels. So it's not just pancreas releases glucagon and boom, we have more glucose in the blood. No, we're going to have a couple of different functions and processes that allow for extra glucose in the blood, like breaking down glycogen. Glycogen is a polymer made of glucose. So if you break off little independent glucoses from glycogen, you can add that to the blood, and now you have more glucose in the blood. That process of breaking down glycogen into glucose is called glycogenolysis. We can also make glucose by synthesizing glucose from lactic acid or other non-carbohydrates such as amino acids, and that process is called gluconeogenesis. And that would also release glucose into the blood. So we can break down a polymer made of glucose, or we can make glucose. But those are two processes, glycogenolysis and gluconeogenesis, respectively, that release glucose to the blood. And that's how we're going to get the effect of glucagon. Now, I like to remember glucagon makes glucose gone from cells. So if it leaves the cells, it has to go to the blood. So we're getting a rise in glucose. Insulin does the opposite of glucagon. So insulin and glucagon are definitely antagonists. They do the opposite of each other as far as function is concerned, and they're both released from the same gland. In this case, the pancreas is acting both as an endocrine and exocrine gland. Insulin is going to decrease blood glucose levels, so it's secreted when blood glucose levels increase. It's usually secreted as kind of a precursor called proinsulin, and then has to be modified. And it's able to lower blood glucose in a couple of different ways. So it can enhance membrane transport of glucose into fat and muscle cells. It can inhibit the breakdown of glycogen to glucose and inhibit the conversion of amino acids or fats to glucose. So we can stop the breakdown of glycogen to glucose. So therefore the glucose can either be put into glycogen, or we're no longer increasing glucose levels in the blood by breaking down that polymer. We can make sure that the glucose that is present goes somewhere else, maybe into adipose tissue or skeletal muscle tissue, for example. And we can stop the conversion of other sources of glucose synthesis, such as amino acids or fats. And we can stop that conversion of those nutrients into that sugar, glucose. So insulin is not really needed for glucose uptake in the liver, kidney, or brain, but it does definitely play a role in neuronal development, learning, memory, and we find that insulin actually binds to its very own tyrosine kinase enzyme receptor, so it doesn't exactly work via second messenger system, even though it's kind of amino acid based. It binds immediately to the enzyme, which we would generally see as the third step in a second messenger system. So it's able to kind of trigger its own protein kinase cascade. Insulin also triggers cells to catalyze oxidation of glucose for ATP production. That's the first priority, is to make sure there is glucose available for ATP production. So to make sure that we can dump that glucose into the right location for cells to use in order to be able to produce ATP as a process in cellular respiration. We want to be able to polymerize glucose to form glycogen and convert glucose to fat, so particularly in adipose tissue. Insulin works in a couple of different ways. Factors that influence insulin release are, of course, elevated blood glucose levels. We talked about that being the primary stimulus, rising blood levels of amino acids and fatty acids, release of acetylcholine by parasympathetic nerve fibers, and hormones. So you see that list here. So glucagon release actually does cause the release of insulin in order to stabilize the amount of glucose in the blood. Somatostatin and the sympathetic nervous system are also going to be able to inhibit insulin release. So here, figure 15.17 is looking at insulin and glucagon from the level of the pancreas and how we can regulate blood glucose levels. So we're looking at kind of a seesaw, and they're showing us an imbalance. In one way, we're going to have an imbalance where we have an increase of blood glucose. And the other way, we're going to have an imbalance that is a decrease of blood glucose. So we'll start over here on the left. If your stimulus is an increase in blood glucose levels, that imbalance will trigger the pancreas to produce insulin. Insulin will act on cells and tell them to take in glucose, and it'll act on the liver and tell it to take in glucose and turn it into the polymer glycogen. And that will get rid of any excess glucose in the blood. So blood glucose levels fall to normal range. If we have a decrease in blood glucose levels, that's going to tell the pancreas to release glucagon. Glucagon is going to stimulate the liver to produce glucose from glycogen by breaking down glycogen, and therefore we'll have blood glucose rise to normal range. And we'll be back at a balance or homeostatic range of normal blood glucose levels, which is about 90 milligrams per 100 milliliters of blood. A homeostatic imbalance associated with insulin and glucagon is diabetes mellitus. So mainly going to be associated, though, with insulin. The hyposecretion of insulin is type 1, the hypoactivity of insulin is type 2, and there are 3 cardinal signs of diabetes mellitus. You should be aware of these: polyuria, polydipsia, and polyphagia. So polyuria is a huge urine output because the excess glucose that ends up in the blood as a result of not enough insulin being produced and or acting is going to cause that glucose to act as an osmotic diuretic, which produces polyuria, or a lot of urine. Polydipsia is excessive thirst, which is from water loss due to the polyuria. And so you're going to be very thirsty if you're suffering from type 1 or type 2 diabetes mellitus. And then polyphagia is excessive hunger and food consumption. So a lot of eating. So phage is going to be food or eating. And that's going to be because cells cannot take up glucose and are starving. And oftentimes diabetes mellitus is called starving in the land of plenty because the cells do have glucose. They are just unable to take up that glucose, unable to take it into themselves and use for cellular respiration and the production of ATP. Another clinical or homeostatic imbalance is when sugars cannot be used as fuel. So as in diabetes mellitus, fats are going to be used instead of sugar, and this causes lipidemia or high levels of fatty acids in the blood. This can be dangerous because fatty acid metabolism results in the formation of ketones or ketone bodies. These ketone bodies are acidic, and so if they build up in your blood, they can make your blood acidic. That condition is called ketoacidosis. It also causes ketonuria, which is acidic urine, or the ketone bodies end up in your urine. Untreated ketoacidosis can cause difficulty breathing, especially increased breathing or hyperpnea, disrupted heart activity, disrupted oxygen transport, depression of the nervous system, and can possibly lead to a coma and death. There's also hyperinsulinism, which is the excessive production of insulin and its secretion. This can cause hypoglycemia, which is low blood glucose levels. Symptoms of hypoglycemia include anxiety, nervousness, disorientation, unconsciousness, even death, and the treatment's just sugar ingestion. I do recommend going over this chart. It's not the most useful for study reasons, but just to go through it, make sure nothing's catching you off guard. Like, for example, what hyperglycemia is, what hypoglycemia is, ketones, keto bodies, ketonuria, polyuria, polydipsia, polyphagia. None of those words should be throwing you off. You should know what they mean. So just kind of look over figure 15.18 to look at the consequences of insulin and its deficit causing diabetes mellitus. Next, we're going to look at the gonads and the placenta. Now, the gonads produce the same steroid sex hormones as those of the adrenal cortex. So we had weak androgen production from the adrenal cortex. We're going to get more production of androgens from the male and female gonads. The ovaries produce estrogens and progesterone, and estrogen will be looked at much more in detail when we get to chapter 26, but estrogen is responsible for the maturation of reproductive organs. It works with thyroid hormone in that respect. It's responsible for the appearance of secondary sexual characteristics, which begin with the release of weak androgens from the adrenal cortex. Progesterone causes breast development and is responsible for cyclic changes in the uterine mucosa. Whereas the testes, which are the male gonads, produce testosterone. This is able to initiate maturation of male reproductive organs, cause the appearance of male secondary sexual characteristics and sex drive. Definitely necessary for normal sperm production. Testosterone is produced from the interstitial cells or Leydig cells of the seminiferous tubules. And with thyroid hormone, testosterone is able to maintain reproductive organs in a functional state and at normal health. The placenta is going to be able to secrete estrogens, progesterone, and human chorionic gonadotropin, or HCG. The placenta is a temporary hormone secreting structure. We also just want to acknowledge that adipose tissue can release some hormones like leptin, resistin, and adiponectin that have functions like appetite control, insulin antagonist, and enhancing sensitivity to insulin. So these hormones to a degree are being studied in order to treat certain aspects of diabetes mellitus. The GI tract or gastrointestinal tract, also known as the alimentary canal, can also produce some hormones. There are cells in the alimentary canal or GI tract called enteroendocrine cells, and these enteroendocrine cells secrete hormones. Entero is a prefix commonly associated with the intestines, and in some textbooks, the entire gastrointestinal tract. Gastrin, ghrelin, secretin, cholecystokinin, CCK, and incretins are all enteroendocrine hormones produced by such cells. So gastrin is commonly known as the hunger hormone. Ghrelin from the stomach does stimulate food intake. Secretin and cholecystokinin activate the pancreas and act on the liver and gallbladder in order to cause its secretion of things like bile and pancreatic juice. And then incretins enhance insulin release and inhibit glucagon production. We've already talked about atrial natriuretic peptide, or ANP, which is secreted by the heart and decreases blood pressure and blood volume by decreasing the reabsorption of sodium into the blood. Atrial natriuretic peptide is an antagonist to aldosterone, so that's when we looked at it before. So if you go back to the chart that looks at all of the kind of triggers for the release of aldosterone, you'll see it there as an inhibitor for the release of aldosterone from the zona glomerulosa cells of the adrenal cortex. The kidneys release renin. We've seen renin before in the renin-angiotensin-aldosterone mechanism. And erythropoietin is probably one of the only hormones on this slide that we haven't talked about. The abbreviation for erythropoietin is EPO. And when we start the blood chapter, we will go into more about EPO or erythropoietin, but it's basically responsible for the production of red blood cells. Even your skeleton can release some hormone-like substances, like osteocalcin from osteoblasts, which acts on the pancreas, tells them to secrete more insulin. It's activated by insulin, and low levels of osteocalcin are actually present in type 2 diabetes. So there is research looking at perhaps increasing levels and how that might be a new treatment for that type of diabetes mellitus. Your skin produces cholecalciferol and calcitriol. Cholecalciferol is a precursor to activated vitamin D. Calcitriol is the active form of vitamin D, and it helps absorb calcium from the intestine, which is important as one of the functions of parathyroid hormone. It's also going to modulate immunity, decrease inflammation, and may act as an anti-cancer agent. Mainly on this slide, just be aware that calcitriol is the active form of vitamin D. And vitamin D is going to help us absorb calcium. So activating vitamin D is kind of an indirect effect of parathyroid hormone released from the parathyroid glands. And not last, and definitely not least, but we're going to look at the thymus in a little more detail than kind of what's presented here on the slide. The thymus is oftentimes also called an immune organ or an immune structure. It's very large in infants and children, but it's going to atrophy or shrink with age. It gets smaller and more fibrous, and it does produce hormones like thymulin, thymopoietin, and thymosins. And all of these hormones act mainly as paracrines. They don't really go all throughout the body and travel far in the blood. So we're kind of loosely calling thymulin and thymopoietin and thymosin hormones. They really stay around the thymus and are involved in the normal development of T lymphocytes. T lymphocytes are called T lymphocytes because they develop in the thymus, and they're responsible for part of the adaptive immune response. So these hormones listed here in bold are definitely classified as hormones, but act more as paracrines, and they act less and less prominently as we age because of the atrophy of the thymus. And that's it for part B on the endocrine system. Stay tuned for the blood PowerPoint videos.